What if everything you were ever taught about the origin of life on Earth was incomplete? Not wrong, just missing the most important piece. What if the answer wasn't buried in a laboratory or hidden inside ancient rock, but traveling through our solar system right now, at 60 kilometers per second, leaving behind a chemical signature that the world's most powerful telescopes can detect, but cannot explain? That object is real. Its name is 3I Atlas. And what it has just done chemically, physically, and scientifically has quietly sent shockwaves through the closed-door briefings of institutions that have never once entertained this conversation publicly. On March 2026, the James Webb Space Telescope, the Spheric Space Observatory, and ground-based spectroscopic arrays spread across 3 continents locked onto 3I Atlas in a coordinated observation campaign with no precedent in the history of planetary science. The data flowing back to analysis teams at the Jet Propulsion Laboratory and the SETI Institute was not producing the chemical fingerprint of a standard interstellar comet. It was producing something the models simply could not account for. And the explanation being whispered in those closed-door sessions, the one that none of these institutions have ever entertained publicly, is the kind that doesn't just update a textbook. It rewrites the story of life itself. Here is what happened. 3I Atlas is releasing methane. But not when it should. Not where it should. The methane only appeared after perihelion, after the heat of our sun had time to penetrate deep into the object's interior. And that single fact, as quiet and technical as it sounds, violates every known model of cometary chemistry in the scientific literature. To understand why this matters so enormously, you need to understand how comets are supposed to behave, and why 3I Atlas is doing the opposite. Cometary nuclei are frozen mixtures of water, carbon dioxide, carbon monoxide, methane, and complex organic compounds locked together with silicate dust, formed in the deep cold of interstellar space. As a comet approaches a star, solar radiation heats the surface, and these frozen compounds sublimate. They skip the liquid phase entirely and transition straight from solid to gas. Crucially, this process follows a strict sequence determined by each compound's sublimation temperature. The most volatile substances go first, at the greatest distance from the star. The less volatile ones activate progressively as heating intensifies closer to perihelion. Carbon monoxide is among the most hyper-volatile cometary compounds, sublimating at approximately -205 degrees Celsius. Methane is slightly less volatile, sublimating at around -182 degrees Celsius. Both should appear early in any inbound observation campaign, long before perihelion, and both should decrease in output as the comet's surface reserves are depleted during the approach. This is not a theory. It is a well-documented, repeatedly confirmed behavior across every comet humanity has ever studied in detail. Spherics detected carbon monoxide from 3I Atlas during its inbound approach. That was expected. That was normal. What was not normal was this: zero methane signal. Across the entire inbound observation window, despite methane being only slightly less volatile than the carbon monoxide that was clearly and consistently present, there was no methane at all. Then the 3I Atlas passed perihelion, and methane appeared. Under every natural comet model that exists, this is chemically impossible. If methane ice were present anywhere near the surface of 3I Atlas, it would have sublimated before carbon monoxide, not after it. The only physical scenario in which methane stays absent during inbound heating and emerges only after deep solar penetration is one where the methane was never present as surface ice. Where it was being generated deep inside the object, in a region that only crossed a thermal threshold after perihelion warmth had worked its way through the outer layers. On Earth, there is one primary process known to produce methane on a delayed thermal trigger buried inside a planetary body. It is called methanogenesis, and it is biological. Methanogenesis is the metabolic process by which ancient microorganisms called methanogens produce methane as a byproduct of anaerobic respiration. These are not exotic, theoretical life forms. They are among the oldest organisms on Earth, members of the domain Archaea, capable of thriving in environments that would destroy nearly every other known life form. They live inside volcanic vents, deep within subsurface rock, in oxygen-free sediments, and across temperature ranges stretching from near freezing to above 100 degrees Celsius. Methanogens produce methane in the intestines of every mammal on Earth. They generate it in the sediments beneath every wetland on the planet. They drive it out of the deep rock of the continental crust, and they operate on a single governing principle. Warmth triggers production, cold stops it, and warmth restarts it. They do not die in the cold, they wait. When the temperature climbs back above their threshold, the methane flows again. The astrobiology framework for interpreting the 3I Atlas methane anomaly is not speculation. It is the direct application of established biosignature detection methodology, the same analytical approach that shaped the design of instruments aboard the James Webb Space Telescope and the Europa Clipper mission. A thermal activation signature for methane production, appearing only after sufficient solar heating has penetrated to a protected interior environment, is precisely what theoretical models predict for dormant biological metabolism reactivating after an extended period of cold dormancy. This concept has a name, and it is not new. Directed panspermia, the hypothesis that life can be deliberately transported between star systems by technologically advanced civilizations, was formally proposed by Francis Crick, co-discoverer of the double helix structure of DNA, in a 1973 paper published in the journal Icarus. Crick's argument was grounded in a specific and deeply uncomfortable anomaly in the genetic record: the universality of the genetic code across all life on Earth. Every organism on this planet, from ancient archaea to modern humans, uses the identical 64 codon system to translate DNA into protein. This universality implies common ancestry. All life on Earth descends from a single origin point. But that origin appeared in the geological record with what Crick himself described as suspicious speed within a few 100 million years of Earth cooling sufficiently to support liquid water. In geological terms, that is barely a blink for a process as chemically intricate as abiogenesis. Genomic analysis of 700 modern microbial species, tracing their evolutionary relationships backward to reconstruct the characteristics of the last universal common ancestor, places that ancestor at approximately 4.2 billion years ago. Earth itself formed 4.5 billion years ago. Life appeared within 300 million years of planetary formation, a time frame that many abiogenesis researchers consider uncomfortably compressed for chemistry alone to produce even the simplest self-replicating system from scratch. We also know that interplanetary transfer of biological material is not science fiction. The Martian meteorite designated ALH 84001, recovered from Antarctica in 1984 and confirmed by isotopic analysis to be of Martian origin, was never heated above 40 degrees Celsius during its entire transit from Mars to Earth. That is body temperature, a temperature at which every known category of extremophile microorganism remains fully viable. Physical proof that rocks can travel between planets without sterilizing their biological contents does not sit in a theoretical paper. It sits in laboratory storage at NASA's Johnson Space Center. If interplanetary transfer is confirmed, and if the methane anomaly inside 3I Atlas is biological in origin, then the question the complete dataset forces into the open is not simply whether life can survive interstellar transit. It is whether the life inside 3I Atlas was placed there deliberately, and whether what just activated near our sun was always meant to arrive here. Deliberately placed life requires three things: a delivery mechanism, a preservation architecture, and a targeting system capable of selecting a destination star system billions of years before arrival. Each of these requirements, examined against the physical and chemical data 3I Atlas has produced, resolves not into impossibility, but into a specific engineering framework that theoretical models of directed panspermia have outlined in peer-reviewed literature for over 50 years. The preservation of architecture is the first requirement. Dormant microorganisms traveling through interstellar space face three primary extinction pressures: cosmic radiation, thermal extremes, and time. Cosmic rays, high-energy charged particles that saturate interstellar space, carry sufficient energy to damage DNA at doses that accumulate across millions of years. Water ice provides partial shielding, but its effectiveness decreases as radiation energy increases. The most radiation-resistant organism known on Earth, Deinococcus radiodurans, officially listed in the Guinness World Records, can withstand acute radiation doses of 5,000 Gray, a quantity that would kill a human at just 5 Gray. It achieves this through extraordinarily efficient DNA repair mechanisms that can reassemble shattered chromosomes from multiple fragments. Theoretical models examining the survival of Deinococcus-class organisms embedded within meters of protective ice or silicate matrix over interstellar transit timescales of tens of millions of years suggest that viability is not excluded. Radiation shielding provided by the surrounding bulk material reduces the effective dose rate to levels potentially compatible with survival given sufficient repair capacity. The spherics detection of complex organic molecules releasing from 3I Atlas at 14% of the water production rate is consistent with a nucleus containing preserved biological material distributed through an ice matrix, exactly the architecture a directed panspermia payload would require. The thermal preservation requirement is addressed by the same structure. Interstellar space maintains temperatures near 3 Kelvin, approaching absolute zero. At these temperatures, biological activity ceases entirely and chemical degradation rates drop to effectively zero. Organisms in this state are not dead, they are suspended. Revivification upon warming is documented across multiple organism categories. Tardigrades, microscopic animals capable of surviving vacuum, radiation, and temperatures approaching absolute zero through a process called cryptobiosis, have been successfully revived after decades of cryogenic storage under laboratory conditions. Bacterial endospores recovered from 250-million-year-old salt crystals in New Mexico have been cultured after rehydration. The scientific literature on long-duration biological dormancy and successful revival is not fringe science. It is a well-established research field whose implications become dramatically relevant when applied to an object that spent billions of years in interstellar space before receiving the solar warmth that triggered methane production. A secondary thermal management mechanism, described in directed panspermia theoretical frameworks, involves radioactive heat sources. Radioactive decay releases energy as heat continuously over geological timescales. Uranium-238 has a half-life of 4.5 billion years. Thorium-232 has a half-life of 14 billion years. An engineered payload containing concentrated radiogenic material embedded within a comet nucleus would generate a sustained, low-level thermal environment in the immediate vicinity of the biological payload, maintaining temperatures above the minimum threshold for metabolic activity even during the coldest stretches of interstellar transit. The late-appearing methane from 3I Atlas's interior, emerging only after deep solar penetration triggered thermal activation, is consistent with a payload geometry where organisms inhabit a radiogenically warmed interior environment, one that maintained metabolic viability throughout the entire transit, shielded from surface radiation by overlying layers of ice and rock. The organic molecule inventory detected by James Webb in 3I Atlas's coma provides the final piece of the substrate picture. Water, carbon dioxide, carbon monoxide, and methane, the four metabolic molecules, identified collectively, constitute the complete resource set for methanogenic archaea. Methanogens consume carbon dioxide and hydrogen to produce methane and water in the most energetically fundamental metabolic reaction known to biology. They need no oxygen. They need no sunlight. The substrate was already there. The warmth arrived at perihelion. And the methane appeared precisely on schedule. Isotope ratio analysis is the measurement that would resolve the biological hypothesis definitively. Living organisms preferentially incorporate lighter isotopes of carbon, nitrogen, and sulfur into their biochemistry through kinetic isotope effects, the slightly faster reaction rates of lighter isotopic variants in enzymatic processes. This biological isotope fractionation produces characteristic ratios of carbon: 1,2 to carbon, 1,3, nitrogen, 1,4 to nitrogen, 1,5, and sulfur, 3,2 to sulfur, 3,4 that are measurably distinct from the ratios produced by purely abiotic chemistry. Meteorite analysis programs examining carbonaceous chondrites have established baseline isotope ratios for non-biological organic synthesis in solar system formation environments. The methane and organic compounds releasing from 3I Atlas carry isotope signatures in the spectral data, but the resolution of current remote spectroscopic observation cannot distinguish biological from abiotic fractionation at the precision required for definitive classification. A physical sample would provide that resolution instantly. We do not have one. 3I Atlas is moving at 60 kilometers per second. Every hour, it travels 216,000 kilometers farther from the Sun, from our telescopes, and from any possibility of physical contact. The observation window during which the spectral signal remains strong enough for high-resolution analysis is measured in months, not years. The spacecraft that could have intercepted it launched within weeks of the July 2025 discovery, using existing propulsion technology on a trajectory calculated to match velocities at the December closest approach, was never built. The decision was never made. The opportunity, which will not recur for this specific object, passed without contact. The single most significant unresolved question in the entire forensic record of 3I Atlas is not whether the methane is biological. The instruments are not yet precise enough to answer that definitively from Earth's distance. The deeper question is whether this object was specifically targeted at this solar system, whether its trajectory was calculated not by the random physics of interstellar ejection, but by an intelligence that selected this star, this timing, and this approach geometry with deliberate precision. And if so, whether the biological payload it may be carrying was intended to arrive at a world that had already developed the technology to recognize what was being delivered. The recognition timing is not incidental, it is the variable that transforms directed panspermia from a philosophical hypothesis into a testable forensic framework. A civilization seeding life across the galaxy faces a fundamental delivery problem. The target world must be biologically receptive when the payload arrives, but it must also be technologically aware enough to recognize a delivery, rather than simply absorbing it as one more cometary impact in a geological record full of them. Too early, and the organisms arrive on a sterile world with no established biology to integrate with. Too late, and the civilization that might have acted on the discovery has either collapsed or moved beyond the window of meaningful response. The optimal arrival window is narrow. A civilization that has developed radio telescopes, space-based spectroscopy, and astrochemical analysis capability, but has not yet achieved interstellar travel. A civilization that can see exactly what is arriving, but cannot follow up with physical contact. A civilization that receives the biological inheritance passively, while simultaneously developing the awareness to understand what it received. We are precisely that civilization. We detected 3I Atlas 7 months after it entered the solar system. We identified its anomalous chemistry within weeks. We recognized the methane biosignature anomaly within the observation campaign. We are writing the forensic analysis of a potential directed panspermia delivery event in real time, while the object that may have executed it exits our solar system at 60 kilometers per second. We can see it, we cannot touch it, we can describe what it may have carried, we cannot yet confirm it. ALH 84001 traveled from Mars to Earth without exceeding 40 degrees Celsius. Life on Earth appeared 300 million years after planetary formation, impossibly fast for chemistry working alone. The last universal common ancestor existed 4.2 billion years ago, and the 3I Atlas appeared in our sky 4.6 billion years after our solar system formed, releasing biological methane signatures precisely after perihelion warming penetrated its interior. These data points are separated by billions of years. They are part of the same sentence. We may not be the first civilization to receive a delivery from the cosmic gardeners. We may be the product of one. And 3I Atlas, now accelerating toward the constellation Gemini, carrying whatever its surveillance collected across 45 days of optimal Earth observation, is filing its report on how the garden turned out. The seeds were planted before we existed. The harvest is us. If this made you think differently about where life comes from and where it might be going, share this video with someone who needs to hear it. Subscribe for the next transmission, because this story is far from over.